Okay, so hi everyone. Welcome to today's Pain Research Forum webinar. We're having a wonderful panel of speakers today to talk about the new developments in descending modulation of pain. So before we get started, I just want to mention a few housekeeping rules. So feel free to ask questions to the speaker and moderator by submitting your question from the Q&A function on Zoom. Oh, sorry. Okay. Uh, and uh, and also Zoom allows you to zoom in and out from the share screen using the view option menu. And this webinar is recorded and the recording will be available later on painresearchforum.org and the PRF YouTube channel. And we have upcoming webinars lined up for next month, a monthly webinar lineup. If you are interested in them, feel free to register using the QR code showing on the screen. So our June webinar is on oral facial pain. And also we have a upcoming journal club on June 16th. That's collaboration with the MASIG IASP MASIG interest group. Okay, I'm gonna stop my screen and I will let Harrison to introduce himself and the speakers.
Thank you so much, Light. And good morning, everyone. My name is Harrison Stratton. I'm a postdoctoral fellow at the Pittsburgh Center for Pain Research. Thank you for joining us today for this webinar hosted by the Pain Research Forum. For this next installment in our series, we're going to be discussing the topic of descending modulation in the context of pain with three very prominent researchers in this field. Beginning our webinar, we have Dr. Frank Porreca, professor of pharmacology at the University of Arizona in Tucson. Dr. Porreca's research focuses on understanding brain circuits that promote pain, migraine, post-traumatic headache, and the overlap of these circuits with opioid addiction. Next, we will hear from Dr. Kirsty Bannister, Associate Professor of Sensory Neurophysiology at Imperial College London. Her team bridges the gap between preclinical and clinical pain research as they conduct exploratory experiments that seek to define functionally the somatosensory processing in humans and animals. Our last speaker today will be Dr. Patrik Ernfors, Professor of Tissue Biology at the Karolinska Institute. His research group studies ascending and descending neural pathways in the somatosensory system that underlie the ability to sense touch, temperature, itch, and pain. So we'll have time for questions at the end, after the talks, and if you could submit your questions via the Q&A function at the bottom of your screen, that would be great. And with that, I'll hand it off to Dr. Porreca, who will give our first talk of the session.
All right. Well, when Harrison and Becky invited me to participate in this event, I indicated that I would do so if I could give a little bit of a historical overview of the concept of descending modulation. And what I'd like to do is to do that and show you a couple of early experiments that came from my lab. And I'd like to leave the new information to Kirsty and to Patrik. So I want to begin with this concept of the relationship between activation of nociceptors and the perception of pain. And so we know that when we activate nociceptors, we almost always produce the perception of pain, but not always. And so that really was the kind of conundrum that was faced by our early investigators. And so this was noticed as far back as 1946. This is Dr. Henry Beecher. He was an anesthesiologist from Harvard who was treating soldiers in the Second World War. And what he noticed is that many of the soldiers that had these grievous wounds denied that they were experiencing pain, refused analgesics. And this was very, very different from what he had experienced in his civilian practice. And so he wrote in his famous paper in 1946, Pain in Men Wounded in Battle, that there's no simple relationship between stimulus and subjective response to pain. And so these kinds of observations have really led us to understand that we can't look at a wound and predict the amount of pain or the degree of pain that somebody's experiencing. This looks like a very serious wound, a little scrape on the knee, of course, no big deal. But again, it really depends on the context in which the nociceptors are activated for the perception of pain. So nociception then is subject to very powerful endogenous modulation. And it was these kinds of observations that led some of the fathers of our field, people like Pat Wall and Ron Melzack, to start to introduce the concept that nociceptive input could be processed, could be modulated at the level of the spinal cord, so that the output of the spinal cord that reached the brain that resulted in the perception of pain was already under some form of control. And so that led to their publication of the famous gate theory of pain control. And I just want to review a couple of the points that they made in their writings. They wrote that the firing pattern of even the most specific cells in the spinal cord is the consequence of the presence of peripheral damage in a specific area, the nature of other stimuli in other peripheral areas. And this is the one that sometimes gets forgotten, the descending control by pathways originating in the brain. And so this combination of factors determines the message ascending from the first central cells and was described as the gate control theory, published by Wall and Melzack first in 1962, but the paper that most people remember is in 1965. And the importance of the theory is that this was the theory that was enabled the sensory apparatus that was described by Pat Wall, the body, to be connected to the mind, the perception of pain. So now we recognize that pain, of course, is a sensory experience, and it's modulated by many factors: attention, distraction, emotion, expectation, stress. All of these can change the consequences of nociceptive input. And so that started to lead to the concept that the brain itself can control, in many ways, the output of the spinal cord that leads to the perception of pain. And here's a nice photo of a young Howard Fields, along with Pat Wall, as these concepts were being introduced. So context, past experience, interpretation all determine the most advantageous decision for behaviors that benefit the survival of the organism. That's really the role of descending modulation. Sometimes we want to respond to pain, but sometimes it's beneficial to suppress pain and to perform some more expedient behavior, such as to escape and to survive. Okay, so the idea that there was a descending pain modulatory circuit arose from many, many different papers and different findings. And I wanted to show you a couple of early papers that started to lead to this concept. As far back as 1971, there was this paper that was published by John Libeskind's group, along with David Mayer. Huda Keel was also an author on this paper, that showed that electrical stimulation of the brainstem of the rat could produce analgesia. And this was followed just a year later by the identification of an opioid receptor by Pert and Snyder in 1972. And so the presence of an opioid receptor in the brain of the rat suggested that there might be endogenous ligands for the receptor, and these were discovered in 1975 by John Hughes and Hans Kosterlitz, the endogenous opioids, the enkephalins. And so now we had some basis for an opioid-sensitive circuit in the brain. And in one of the most remarkable rapid translation of basic science that really may have ever occurred, this paper was published by Hosobushi and colleagues that showed that pain relief could be achieved by electrical stimulation of the central gray matter in humans. And most importantly, it demonstrated that it was reversed by naloxone. So it really suggested the presence of an opioid-sensitive circuit in the brain that could modulate pain. And so since that time, there have been studies on deep brain stimulation where there have been successful relief of pain in humans. Okay, so how does it work? And so for this, we turn to the seminal work of the laboratories of Alan Basbaum, who did some critically important anatomical studies showing that cells in the rostroventral-medial medulla could descend to the spinal dorsal horn, and electrophysiological studies from Howard Fields, from the laboratory of Howard Fields, that classified cells in the rostroventral-medial medulla. And so the genius of the work of Howard and his colleagues, Mary Heinricher, Ian Meng, and others, was to characterize the firing of these cells in response to a noxious stimulus, pairing the electrophysiology with a behavior. And the behavior was the simplest behavior possible, with the tail flick. And so that allowed Howard and his colleagues to identify cells that would respond to the noxious stimulus. So some cells in the rostroventral-medial medulla were firing, and they turned off when the noxious stimulus was applied and the tail flick occurred. Other cells were silent; they started to fire when the noxious stimulus was applied and the tail flick occurred. And so the off cells were classified as pain inhibition cells. The on cells were classified as pain facilitation cells. And so then the group went on to look at the consequences of analgesic drugs. Drugs such as opioids would increase the firing of the off cells, the pain inhibition cells, and suppress the firing of the on cells, the pain facilitation cells. And these were all concomitant with inhibition of the tail flick. And so again, this concept then emerged that this was an opioid sensitive circuit in the brainstem that could modulate pain, and it was bidirectional. And so a variety of other studies then started to identify where receptors might be expressed on these cells. And what became apparent is that the pain facilitation cells, the on cells, would express the mu opioid receptor. So these cells might be characterized by expression of the mu opioid receptor. Now, at that time, of course, people were interested in other types of opioids and opioid receptors. And so kappa receptors were thought to be potentially analgesic. But when they were microinjected into the brain, they produced hyperalgesia. And so the concept emerged that the off cells, the pain inhibition cells, might be the cells that express the kappa opioid receptor. And so mu and kappa opioid receptor expressing cells might have opposing actions within the rostral-ventral median medulla. And this seemed to be true and likely is true in other brain circuits as well. Okay, so all of those studies were done in modulation of acute pain. And around this time, my laboratory was interested in understanding how these cells might participate and how descending modulation might participate in chronic pain. And the way that we approached it was to use one of the interesting tools that was available at that time, and that was a ribosome inhibitor called saccharin. So the idea was that if we conjugated saccharin to a mu opioid receptor agonist, such as dermorphin, highly selective for the mu opioid receptor, that the conjugate, the dermorphin saccharin, would be internalized. The G protein coupled receptor would essentially be a portal of entry into the cell, and this would prevent protein synthesis and would kill the cell. And so this was sort of a surgical deletion of cells in the rostral-ventral medial medulla that expressed the mu opioid receptor. And so we did this experiment. It was a prevention experiment. We first injected the dermorphin saccharin, and then we waited 28 days, and then we performed spinal nerve ligation. These studies were done in rats. I'm showing you the tactile allodynia, but the thermal hyperalgesia is exactly the same. And so what you see here on the slide is that animals that undergo spinal nerve ligation will show sustained tactile allodynia. This is very typical. But if we lesion the cells in the rostral-ventral medial medulla that express the mu opioid receptor, you see that they develop pain initially, but they don't sustain the pain. And so the chronic pain seems to be reversed. And so mu opioid receptor expressing cells seem to be required for the expression and the maintenance of neuropathic pain. And we interpreted that as suggesting that it was due to enhanced descending facilitation, presumably from mu opioid receptor expressing cells in the RBM that would be putative on cells. Okay, so more recently we have more advanced tools. And so we had mice in which we could express a GI dread into mu opioid receptor expressing cells in the rostral-ventral medial medulla. We performed partial static nerve ligation. And then we gave the dread ligand CNO by different routes. So we gave CNO intraperitoneally. And you can see that that produced a transient reversal of the pain behavior. Then we administered CNO in the drinking water, and you see that there was a sustained reversal of neuropathic pain, established neuropathic pain, and the pain response came back after we took the CNO away. And then we gave an intrathecal injection of CNO, and again, we saw this transient reversal. So targeting the terminals, presumably, of the mu opioid receptor expressing cells in the RBM. And so again, we interpreted these as on cells, pain facilitation cells that would be required to sustain and maintain, in this case, established neuropathic pain. Again, these descending projections then would be the cells that would determine the output of the spinal cord to the brain that would lead, presumably, to the perception of pain. Here's another early experiment that we did. We were also interested in the kappa opioid receptors and their potential role in modulating the descending pain inhibition cells. And so what we had at the time, what was available to us at that time was a pro-dynorphin knockout mouse. It was a global knockout. And so if you look at this image on the left, you see that it looks exactly the same, really, as what I showed you with the dimorphin saccharin. We performed the spinal nerve ligation. Animals that make dynorphin and have sustained pain. Animals that don't make dynorphin will reverse the pain. And so we interpreted this as suggesting that there was a time-dependent upregulation of dynorphin in the rostroventral median medulla that would inhibit descending inhibition. And so dynorphin signaling might be required for the maintenance of chronic neuropathic pain. Now, of course, it was a global knockout. We actually didn't know where the dynorphin might be producing its effect. And so more recently, with more advanced tools, Sarah Ross and colleagues have used mice in which they could express DREADs in kappa opioid receptor expressing cells in the RVM. Activation of those cells with a GQ DREAD would produce an inhibition of pain. Inhibition of those cells with a GI DREAD would enhance pain. So chemogenetic activation of putative off cells inhibits pain, while inhibition facilitates pain. Okay, so the last little thing that I'm going to tell you is this idea about bidirectional control of nociception and the possibility that really it's the balance of facilitation and inhibition that may be important for the net effect that we might see on pain. And so we were a little bit puzzled that when we did peripheral nerve injury to our animals, we almost always saw so-called neuropathic pain in the animals. But this was different from what is seen in humans. In humans, injuries to nerves will, of course, produce chronic pain in some people, but relatively rare based on which nerve is injured. Maybe about 10% of humans would develop chronic pain. So we went back and looked at our records, and what we found is that when we produced spinal nerve ligation injury in our animals, almost always we got neuropathic pain, but occasionally there were some animals that didn't show signs of pain. And so we went back and looked at our records with surgeries that were performed by a single technician, Janice Oyarzo. And so over the years, we had performed 783 spinal nerve ligation injuries in our Sprague Dawley rats. And what we found is that 85% of these animals were allodynic, but 15% were not allodynic. And then we became aware of another strain of rats that that was being investigated through conversations with the laboratory of Tony Yaksh. These were Holtzman rats. These were rats that sort of are a cousin of the Sprague Dawley rat. And so we did some experiment with these animals, 185 of these animals. These animals were thought to be resistant to neuropathic pain. And in fact, we found that about 50% of these animals were classified as allodynic, injured, but about 50% were injured, but not allodynic. And so then we did this experiment where we took the allodynic animals and injected lidocaine in the rostroventral median medulla, and you see that there was a transient reversal of the pain. So that was to be expected. And so we interpreted that as pain facilitation, inhibition of pain facilitation. But then we took the injured but not allodynic rats. We confirmed post hoc that these animals were injured in the appropriately. And if we looked at peripheral markers, we could see signs of peripheral nerve injury, ATF3, et cetera. And then we microinjected lidocaine into the rostroventral median medulla or a kappa agonist. And what you see is that these non-allodynic injured animals now showed pain behaviors. And so we interpreted that as descending inhibition from the rostroventral median medulla, protecting against neuropathic pain. And so we concluded that the balance between descending facilitation and inhibition is disrupted in chronic pain to result in net descending facilitation. And this is something that I think can be evaluated and has been evaluated in humans using conditioned pain modulation. And maybe Kirsty will talk about that in the future. The last little thing about this is that we also did learning experiments with these animals. So injured allodynic animals, where we put lidocaine in the rostroventral median medulla, would show conditioned place preference. Animals that were injured but not allodynic would show conditioned place avoidance. So this, again, supported this concept. And so the very last thing I'm going to tell you is that we've been talking about bidirectional control in the context of pain. And pain is a motivation, but there are many motivations in life that are competing. And so sometimes it's important for us to deal with the motivation of pain within the context of other overriding motivations, such as rewards. And so it may be advantageous to suppress pain in order to gain rewards, such as food. And so I point you to the motivation decision model of pain that was published by Howard Fields. We don't have time to talk about it, but I think it's a very important part of how we interpret descending modulation. And so this is a last quote from Ron Melzack, where he wrote that it is essential to remember that stimulation of receptors does not mark the beginning of the pain process. Rather, stimulation produces neural signals that enter an active nervous system, which is already the substrate of past experience and culture, anxiety, and so forth. And so again, top-down modulation is what allows us to connect the sensory apparatus with the mind. And I'm going to stop there and turn it over to back to Harrison.
Thank you so much, Frank. Very, very informative talk. And thank you so much for presenting that data. And we'll move right now next into Dr. Kirsty Bannister's talk, who will move on from the historical context and talk a little bit more about some more modern findings. And, thank you, Frank, for that wonderful background. So, Kirsty, if you could unmute and yeah.
Okay, ready. I think.
Take it away.
Great, thank you. Thanks, Harrison, for asking me to talk, and what a pleasure to be on a panel with Patrik and Frank as well. So I'm going to this flows beautifully from where Frank was, to just talk a little bit about how these circuits differ, but most focus there being on the neurophysiology of brainstem modulatory circuits, in order that we can understand the reality of clinical application of translational approaches to assessing neural pathways of the DPMS. We need to understand the function of these circuits, for example, their pharmacology, in order that any translational approach we do can have, in the end, clinical meaning. So the breadth of circuit diversity isn't captured by diagrams like these, but the key loci that are investigated are shown alongside the terminology for these projection pathways and associated pharmacology. And of course, we know that there are these bottom-up mechanisms that are driven by sensory inputs that recruit brainstem circuits and are independent of cortical control, versus top-down mechanisms that are driven by those cortical inputs to converge on brainstem circuits and ultimately determine how our cognitive state can affect pain sensitivity, precisely because they have an impact on spinal neuronal transmission processes. I've just realized that, is this bar right in the way of the titles at the top of the slides?
No, we can see everything.
You can see everything. Okay, great.
Yeah.
That's just me then. Okay, so the of course, for the modulation of dorsal horn neuronal activity is receptor mediated, and that receptor effect will depend on precise expression patterns and organization of microcircuits that might impact primary afferent terminals, interneurons, projection neurons, etc. So quite how that happens depends on the neurophysiology of the circuit in question. And I'm going to just focus a little bit on the neurophysiology of the periaqueductal gray and the RVM, as Frank mentioned, but also bringing in the locus coeruleus, is these really important brainstem circuits that are going to play an important role in how we interpret the translation of paradigms that evoke activity in the DPMS. So the PAG, of course, is this hub nucleus that can activate descending facilitation or inhibition, a crucial site for opioid-induced hyperalgesia, and a number of previous studies demonstrated that glutamate and GABA play these critical opposing roles in terms of nociceptive processing in the ventrolateral PAG, but the inability to exclusively manipulate subpopulations of neurons therein had prevented the solid understanding of the nature of these roles, and this is why this paper was so important, as it was able to demonstrate the different contributions of genetically defined glutamatergic and GABAergic PAG neurons in nociceptive processing, really bringing us so up to date from from those early seminal studies that Frank presented. And this paper in particular was giving us this direct experimental support for a model in which excitatory and inhibitory neurons in the PAG could bidirectionally modulate nociception, something that we knew about all those years ago, but here we've got this neurophysiological understanding of those processes. So then we can apply the same thoughts to the RVM, and the neuronal types present have been molecularly identified. I'm sure Patrik will speak more to this, of course, but their clustering revealed these distinct glutamatergic, serotonergic and GABAergic neuron types. And of course, we know that we've got these, Frank has outlined perfectly well, these on and off cells that impact the activity of these processes. And Mary Heinrich recently showed that actually they're sensitized to visual light in persistent inflammation, outlining not only the potential mechanisms by which the functionality in sensory systems combine, but also actually mechanisms that can underpin the difference in their activity in different pain states. And ultimately, what we want to be able to do is map functional classes to molecular identity of cell types within the RVM, the PAG, the locus coeruleus, in order that we can understand their neurophysiology better. This paper uncovered an RVM spinal cord primary afferent circuit controlling pain thresholds, where the unexpected finding was that chemogenetic inhibition of GABA positive RVM neurons revealed this tonic modulation of spinal nociception via activation of spinal GABA and encephalinergic neurons. So these RVM GABAergic neurons will facilitate mechanical pain by inhibiting dorsal horn interneurons, and really interestingly, these interneurons are gating sensory inputs and controlling pain through these enkephalin and GABA mediated presynaptic inhibitions of somatosensory neurons. And so all of a sudden you can understand about how maybe you could target these to alter pain thresholds. So again, it's just taking all of that foundation information we had from those super important studies Frank outlined and using these really cool technologies in order that we can better understand the neurophysiology of those circuits. So then that brings me to the locus coeruleus, the last one I'll talk about in terms of that broad level neurophysiology. And of course, we want to understand the neurophysiology so that we can better identify pathophysiological features. For the LC, it's Tony Pickering and colleagues who really spearheaded the process of us understanding the features of those neurons. They implemented these targeting strategies using vectors that express channel rhodopsin to produce the stable transduction of these noradrenergic neurons. We could then reliably opt to manipulate them, and discrete units of the locus coeruleus, which has this module organization, could be targeted. And Esther Baracosa and colleagues recently showed that in conditions of neuropathic pain, for example, you see these irregular firing patterns for spontaneous activity in both the ipsilateral and the contralateral locus coeruleus, and the authors postulated that this effect reflects enhanced activity in ascending pain pathways. And so again, we're starting to understand better than ever these neurophysiological processes that underlie those pain modulatory outputs that we observe, in our experiments. We hopped on the back of this wonderful process, being able to label and understand the functional modular organization of the locus coeruleus in a series of experiments where we would activate either the locus coeruleus as a whole or a coeruleospinal unit. And of course, we showed that if we activated either of these modules, we could inhibit spinal neuronal excitability, and then we were able to show how this was mediated pharmacologically via alpha-1 adrenoceptors, but not alpha-2 adrenoceptors. And this then led us to question, we'd got data 11 years ago with Tony Dickinson that showed that the underpinning of a pain inhibits pain pathways noradrenergic, but that it was blocked by alpha-2 adrenoceptor antagonism, which was not what we were seeing here. And actually, really unexpectedly, if we activated this locus coeruleus spinal cord module, we abolished the expression of this so-called diffuse noxious inhibitory control, which even was fundamentally underpinned by the same noradrenergic processes. But this seemed to be suggesting that the locus coeruleus was perhaps, there was a reciprocal governance between the expression of this particular pain inhibitory control and the locus coeruleus, but quite what that was, we didn't know. And so we next went on to delineate what that critical brainstem relay was in terms of mediated diffuse noxious inhibitory controls. We interrogated with those wonderful systems that Tony Pickering had built the foundation for, activity in the A5, the A6, otherwise known, of course, as the locus coeruleus in the A7 nuclei, to really try and understand how their activity had an impact on this pain inhibits pain phenomenon. And we were able to outline that actually it was the A5 neurons that give us this relay for inhibition of spinal neuronal processes via a descending modulatory pathway whose final relay is the A5. Okay, so having thought then really briefly and broadly about the neurophysiology of some of these brainstem pathways, we'll now think about how we can use translational approaches to assess pathways of the DPMS. And I think it's really important to understand their underpinning pharmacology so that we can gauge exactly what these pathways are telling us when we apply our translational approaches. I'm going to start with some unpublished work that we've been doing looking at offset analgesia and onset hyperalgesia. And what we see here is that we get a pain intensity rating that maps to a constant temperature, for example, of 47 degrees. You can increase that temperature by a degree. There's a big increase in pain intensity reported by the wakeful human. And then if we have that stimulus offset of a degree, you get this disproportionate decrease in the rating of pain that an individual is reporting. Conversely, for onset hyperalgesia, when we decrease that, let me try and maybe this will be a bit easier for you to see. If we have, again, this baseline 47 degrees application of temperature, we decrease by a degree the temperature here, we get this lowered pain intensity reported by the human. Then on stimulus onset, you get this exaggerated increase in terms of what that pain intensity is then reported as. We back translated that paradigm to get the neural correlate of this, looking at firing frequencies of spinal neuronal responses. And again, what you see is on stimulus offset, you get this exaggerated reduction in neuronal responses as opposed to on stimulus onset where you get the converse. So I moved institute about 14 months ago. My lab, animal research lab, is just about up and running again because we've had to build that from scratch. We have a new lab to go, and now we're collecting the pharmacology for this data. So we hope that this can be published soon, because really interesting what we've observed is that this seems to be underpinned by serotonergic processes. If we antagonize 5-HT7 receptors, we no longer see that decrease in spinal neuronal firing that we observed on stimulus offset in our animals. Whereas if we antagonize the facilitatory 5-HT3 receptor on ondansetron, we lose that upon stimulus onset, that exaggerated increase in spinal neuronal firing that accompanies the paradigm. So this is a little bit watch this space in terms of what's coming next. What we know more about and what there is lots of publications on are these paradigms that we can use to evoke descending inhibition upon use of a conditioning stimulus. So we've got lots of data on DNIC from those early studies that Labars and Dickinson, Besson, etc. did, where we're looking at a neurophysiological endpoint as opposed to descending control of nociception. These are in wakeful animals. We might use capsaicin as a conditioning stimulus, heat as a test stimulus, and we've got this reflexive endpoint in the wakeful animal. And then, of course, conditioned pain modulation, which is a perceptual endpoint that's subject to cognitive influences because the individual is awake when we apply these tests. We showed some years ago that we've got this really nice translational approach to measuring diffuse noxious inhibitory controls. So if you look in orange here, this is an individual who's having these cuff pressure devices inflated, and over time, when the presence of a conditioning stimulus is there, what you see is the individuals can stomach the pressure for longer, they rate the pain lower, and we already showed that we get these really nice neuronal correlates of those perceptual outcomes in our animals when we apply identical testing conditioning stimuli, just like we do with the offset analgesia and onset hyperalgesia. So we have very nice translational paradigms that can allow us to interrogate the spinal neuronal processes that underpin those perceptual outputs in humans. This was a paper we published recently looking at data from multiple participants using a completely standardized protocol. We had over 100 male and 200 female participants, and we had a look at between session changes in responder and non-responder statuses for conditioned pain modulation. I'm just showing the data for pain detection thresholds here. We did it for PTT as well. And you can see here that 27 people on test retest switch status. There's minimal concordance between who's switching status between pain detection and pain tolerance threshold measures. Robust group level CPM effects were observed, but by contrast, on test retest, the reliability of CPM effect magnitudes and responder classifications were poor. And I think this leads to a really important question when we're applying these tests, whether it's in an academic experimental setting where there's a real value to these tests versus a clinical setting. And that is, are these CPM effects dominated by dynamic state-dependent processes, which likely affect the presentation and/or detection of underlying stable trait characteristics. Finally, I'll end on this slide, which was another translational approach we've used to see what the impact of modulation on hyperalgesia is. Again, we designed experimental paradigms that we could apply in humans, looking at high frequency stimulation to evoke these sensitized states and in animals, and apply these conditioning stimuli to see whether evoking modulation actually has an impact on the perceptual and neuronal outputs of these tests. And what we find is that application of a conditioning stimulus to evoke activation of those pain inhibitory processes during high frequency stimulation didn't alter perceived hyperalgesia in humans, and nor did it impact the development of neuronal hyperexcitability in rats compared with high frequency stimulation alone. So we found that this excitatory signaling exceeds inhibitory controls during application of these experimental paradigms, and that really supports targeting of, in this scenario, excitatory signaling to prevent the development of sensitization. Inverting these traditional translation processes in this way has really the potential to provide insight into pain mechanisms, but also validation of therapeutic approaches and optimization of treatment algorithms. So this is important translational work that we'll keep pushing forward. So I hope that by discussing these pathways and their neurophysiology and what the translational approaches are, you're somewhat up to date with just some of the ways in which we can interrogate the descending pain modulatory system. Thank you.
Thank you so much, Kirsty. That was awesome. I really appreciated your insight on the translational element between human and animal studies. So very excited to see that data. And with that, one reminder to the audience, please, if you have questions along the way, submit them via the Q&A function, and we'll get to them after Patrik's talk. We have a couple there now, but would be delighted to have more. And so with that, Patrik, if you could share your screen and unmute, we would be delighted to hear your talk. Brilliant.
There, do you see it?
1 second. Yep, now it's there. Beautiful. We can see it.
Okay, thanks for the kind invitation. I'm really happy to be here and to present together with Frank and Kirsty. And we got into this field relatively recently, and we were interested in understanding, well, the descending pain pathway and how it is affected and the cellular basis for how cognition and emotions control pain perception in the descending pathway. And of course, as we have had the previous speakers have explained the pathway, and we focused on the RVM because it's the last relay structure of the brain that projects to the spinal cord, and in the spinal cord modulates transmission in the ascending pathway. And I won't have time to go through a lot of data, so I'm going to focus on the strategy, technological strategy that we use, and basically summarize our results. So we had the idea, or which is, I guess, not unique, but that behavior is created by molecularly defined neuronal types. So our idea was to create a single cell RNA sequencing atlas of the rostral ventromedian medulla. So we now know exactly molecularly defined neurons in the RVM. And then based on activity-dependent gene transcription, using trap mice, such as the ARC trap in this case, to permanently capture neurons that are active during behavior. And then, once you trap them, you can use that trapping both to identify the cells by sequencing and placing them into the atlas that we have generated, as well as also link the activity of that ensemble to behavior and also to connectivity using chemogenetics or optogenetics. And we also make use of two activity-dependent gene expression trap strategies. I mean, we use the ARC trap, but also FOSTVA mouse strategy, which is called the CANE technology developed by Fan Wang. And using these two, we can actually look at the causality of connectivity between two structures. So we can trap, for example, in the RVM, and then we can synthetically activate those and capture the neurons in the spinal cord. And now using FOSTVA, the TVA, and then we can in those animals then express, for example, Q and GI dreads, so we can activate one and inhibit the other to find causality of that circuit. So here is first the atlas of the RVM that we generated. And looking at the excitatory and inhibitory cells, this is now spatial transcriptomics in the RVM. You can see that there are quite a lot of neurons there. And there are both excitatory and inhibitory neurons in the RVM. And then now we can also place in the different cell types into the RVM, the exact molecularly defined cell types there. So we've done the same for the spinal cord. So the RVM projects to the spinal cord. We dissected out the dorsal horn and molecularly classified the neurons there to create the taxonomy of dorsal horn neurons. And using that information, this is some time ago, we did spatial transcriptomics and found that the different, mostly the excitatory neurons, showed a very distinctly layered presence in the spinal cord. So basically, the spinal cord is built by a layering of molecularly defined excitatory neurons that then links, because primary afferents terminate the specific lamina, and that termination then is reflected by presence of molecularly defined excitatory neurons in the spinal cord. So here is just a brief recap. So in the first experiment, so the idea was then to just generate the atlas, capture neurons, place the neurons in the atlas to understand exactly which kinds of neurons become active, and then link that to behavior. And we thought we would start with morphine, not least because, as you heard Frank tell you, there's been a lot of work on morphine, and we knew that morphine would be increasing activity of off cells, reducing pain through that pathway. So what we did here was we injected a GQ dread and GI dread into RVM, waited 2 weeks, and then we applied heat or morphine to the animals. And at that time, we captured the expression of these dreads in the neurons that become active by heat or by morphine. And then 1 day later, we activate and inhibit the previously activated RVM neurons. And when we did that for heat, we found that activation of neurons that had become active previously by heat in the RVM, activation of only the neurons in the RVM now, leads to a marked drop in von-Frey thresholds. However, inhibition didn't do much, suggesting that these pronociceptive ensembles in the RVM that are activated upon heat are not continuously active. They just become active during the pain, application of pain. However, when we reactivated the ensemble of neurons that became active by morphine, you can see here that we markedly increased the von-Frey threshold. And when we inhibit now in the same animal, the animals became the hyperalgesic or allodinic in this case. And the effect of activation recapitulated that of morphine, suggesting that by just activating RVM neurons is sufficient to cause antinociception to the similar degree as morphine does. But the question is the RVM neurons themselves required for all of the effect of morphine antinociception? And here is what we did to look at that. We reactivate these neurons again, and at the same time inhibit these neurons. And then you can see here that morphine has basically no effect at all. So the RVM ensemble activated by morphine is absolutely critical for morphine antinociception. So now we captured the neurons that were activated by morphine and single-cell RNA sequenced them. So we placed them back into the atlas, basically. And when we did that, we found that 4 cell types showed differences in capture. 2 cell types increased capture, and 2 cell types we found a decreased capture. And we focused on these GLUT2 neurons here, because those neurons, I'm not showing you that, but they had the strongest projections to the spinal cord. And those GLUT2 neurons expressed BDNF. So we got an BDNF Cree animal, so now we could just manipulate activity of those RVM neurons expressing BDNF, which are then the GLUT2 neurons. And when we did that, inhibit those, morphine had no effect in the animal. And then we also used them to identify spinal cord neurons that become active by these neurons, and found that those were inhibitory neurons expressing galanin, which is a specific subtype of GABAergic neurons in the spinal cord lamina 2. And those neurons in turn project and inhibit the ascending pathway, projection neurons in the ascending pathway. And the interesting thing, as I already showed you, that now when we inhibit these RVM BDNF neurons, we get much greater pain response. So meaning that these neurons are continuously active and continuously suppressing pain in the animal. And it's doing so through these spinal cord GAL neurons. So what opioids actually are doing, and in this case morphine, which we tried, is just hijacking this pathway, activating these RVM BDNF neurons, which are glutamatergic off cells, that then activate the inhibitory spinal neurons, which in turn inhibits the ascending pathway. So we were also studying at the same time as the ascending pathway, Ming Dong in the lab, to try to understand the pathway that confers mechanical and heat pain. And we found that in the spinal cord, there is a particular excitatory 21 molecularly defined cell type that confers mechanical nociception. And when we inhibit those, the animals no longer respond to pressure pain. And we found that another cell type, excitatory 19 in the spinal cord, that is also a projection neuron, is required for heat pain, C-fiber heat pain. And the interesting thing was that in that study, Ming Dong found that there is also an inhibitory neuron, inhibitor 8 neuron, that actually is activated by both heat and mechanical stimulus. And that is a feed-forward inhibitory neuron in the ascending pathway that reduces activity of excitatory 19 and 21 neurons. And we did the rabies tracing here and found that the inhibitor 8 neurons are innervated by A-delta nociceptors and A-beta field LTMRs. So this is probably a gate neuron that Frank was talking about. And the interesting thing was in this project that this inhibitor 8 neuron is actually the spinal cord GAL neuron. So basically these RVM BDNF neurons are intersecting in the ascending pathway through these feed-forward inhibitory neurons in the spinal cord. So Muge Altinkokin, a PhD student in the lab, has now been working on sleep deprivation and pain sensitivity. And we know that the consequences of sleep deprivation is increased acute pain intensity perception, and also a decrease in pain thresholds. And in some chronic pain conditions, it also worsens pain intensity. And there is very strong data showing that sleep deprivation is a risk factor for chronic pain. And there are quite good animal models for this. And here's one I'm showing you one here that if you sleep deprive mice, pain sensitivity increases. So we used one of those models and could recapitulate that. We kept the animals awake during their sleep cycle, and this led to a marked drop in the von Frey threshold, as you can see here. And on the bottom, maybe I should use the pointer here. Do you see the pointer now?
Yeah, we can see it.
Well, it's not the perfect pointer. Well, actually, I did the wrong thing. Well, at the bottom, we now capture these neurons with GQ that are active during sleep deprivation and synthetically reactivate these. And we can recapitulate now sleep deprivation-induced sensitization. So, sorry, I don't know what I did now. There it comes. So now we used a very similar strategy to the previous study. We trapped the cells with tomato and sequenced those neurons and could identify that there was an inhibitory neuron type, GABA3 subtype that expressed NPY2 receptor that markedly dropped in activity upon sleep deprivation. And we got the NPY2 or Cree animals, so now we can manipulate only those inhibitory neurons in the RVM. And the interesting thing here is that when we inhibit these neurons, von Frey threshold drops markedly, but when we activate them, nothing happens. And this is in the normal animal without sleep deprivation now, which was very interesting because now what we did now was that we sleep deprived these animals, and then we activated them and we could reverse sleep deprivation-induced allodynia here. So this suggests that these neurons are normally active without sleep deprivation, but upon sleep deprivation activity drops. So now that when we activate them after sleep deprivation, we reverse that sensitization. So I didn't show you, but these are local inhibitory neurons in the RVM. So there are interneurons there, and they connect to pronoceceptive projection neurons that project to the spinal cord to intersect with the ascending pathway. And as I already alluded to here, these neurons are decaying in activity with increasing sleep pressure. So we quantify these, of course, during sleep deprivation, and we think that basically that the activity of these neurons drops during wake time, and that sleep deprivation is just a continuation of that decay in activity. And if that is true, then we should be able to sleep deprive animals and then let them have a short rest, a nap of a couple of hours, and we should reverse the activity of these neurons. And that's what we see, as you can see here in the graph on the right, that we sleep deprive animals, the number of ARC positive neurons drops, and then we, after a 2-hour nap, it returns to control animals. So here is what we think is happening, that these neurons are continuously suppressing a pronoceceptive output from the RVM, which are the green cells. And that's the normal physiology of these cells. They're just continuously active, but upon sleep deprivation, activity drops in these cells, releasing the brake of the pronoceceptive output that now increases the ascending pathway. And I didn't show you that, but we've done this also for inflammatory and stress-induced hyperalgesia. And again, we find that the activity of these neurons drops, and which results in increase in pronoceceptive output. I should say that these neurons are not involved in neuropathic pain. So when we do spare nerve injury, these neurons have no effect at all on that. So here is the acknowledgement. Muge did all the work on the sleep deprivation. Michael Fatt did the work on morphine, and Ming-Dong Zhang on the spinal cord, and got help from a number of people in the lab. And the morphine study was a collaboration with Per Svenningsson, Yunting Yang, and the spinal cord, Hans-Ulrich Seilhofer and Joel Albisetti, and also Malin, Kajsa, Mikael, and Katarina at Uppsala University. Thank you for your attention.
Thank you so much, Patrik. That was very exciting data. And we have a number of questions from the audience. Thank you to all the speakers for being with us today and for hanging on. And thank you also to the audience for hanging on. We're going to get to these questions, the earliest ones submitted, and then go through them one by one. So from an attendee, great presentations. What is the role of dopaminergic signaling in chronic pain, in your opinion? This one wasn't directed towards anyone, so I don't know if anyone wants to take a stab at that.
I'll leave it to Frank.
It's a complicated question. There is a descending dopaminergic projection to the spinal dorsal horn. So so that, you could think about it from that point of view. But the other way to think about dopaminergic signaling in chronic pain is the modulation of the rewarding aspects of pain relief that are associated with the dopaminergic transmission in the mesolimbic pathway. So and I should point out that these cells, the dopaminergic cells in the ventral tegmental area, not only project to places like the nucleus accumbens, but they have a strong projection to the anterior cingulate cortex, where activation of dopamine D1 receptors will produce condition place preference only in injured animals. So I think activation of dopaminergic cells in the cortex can also change the aversive qualities of pain.
Thank you for that answer, Frank. Great one. And then a question from Dr. Michael Gold for Kirsty. So interesting data, Kirsty. Thank you. You've nicely demonstrated it is possible to pharmacologically dissociate components of the offset analgesia and hyperalgesia. The 5-HT effects appeared to be restricted to the second phase of the response. He says, I apologize if you described it, but has the pharmacology for the initial increase/decrease in perception also been identified?
Thanks for the question, Michael. No, it's certainly not by us. And actually, because we want to see what's modulating that effect, that might be a bit of a disaster if we didn't get that first initial increase. So quick answer, no. I'm afraid not. Not with the pharmacologies we've used to interrogate this, which have been noradrenergic and serotonergic.
Gotcha. Awesome. Thank you, Kirsty. And then I believe this is a question for Frank. So differences of chronic pain development between two rat strains are surprising and amazing. Are they related to the genetic background of the animals or any other reasons that you suspect? And if you'll revisit these findings using current research techniques, such as sequencing, do you expect to find any sort of like pain-related genes that might be associated with this difference?
Yeah. Well, I agree. It's amazing,
Yeah.
But I mean, I think it speaks to the fact that we have this remarkable disconnect between our preclinical models and what's actually seen in humans. If we got neuropathic pain every time we injured a nerve in humans, I mean, there would be no surgeries that would ever happen. So the interesting animals really are the ones that are injured but don't actually show the chronic pain. But with strains such as Sprague-Dawley rats, there's just not enough animals to actually study those. The Holton rats were actually derived from Sprague-Dawley rats. So probably over many generations, there's been some sort of genetic drift that has happened. So I don't know what the difference is, but this is where Patrik could actually answer the question actually. right?
Yes.
So that would be an interesting experiment, because it's true also, not only of this strain of rats, but I think that there are strains of mice that are resistant to the development of chronic pain. And so I think it would be quite fascinating to look at animals that have injuries clearly defined in the periphery, where you could look at the expected changes in the primary afferent neurons, but don't actually show the pain behavior. And there, again, these descending modulatory mechanisms that you are uncovering, Patrik, you could try to really uncover what differences might be. So just a suggestion. I mean, you have all of the technology to do it.
Thanks.
Yeah. Great thought, Frank.
As if you need extra work, Patrik. But anyway.
And this second question, there's actually two questions now, if you don't mind, for Dr. Ernfors. One from Erica Harding saying, with respect to the sleep deprivation study, this is really fascinating. To what extent do we need to consider that many, if not most, studies of pain behavior are sleep deprived as they're done during the day, the normal sleep cycle for rodents?
Well, I'm not sure that they are sleep deprived, but I mean, because even in when we do these pain tests, when we put them on the mesh and do one fray, they basically go to sleep while waiting for it to happen.
Indeed.
So I mean, they're not sleep deprived, but I mean, what we've been trying to work out, and there is a study by John Wood. Showing that there are different pain thresholds in the morning versus midday and late afternoon. And we've tried to recapitulate that, but to be honest, I mean, we have failed. We haven't succeeded doing that. Because the idea would be if our neurons decay in activity over the day, we should see a difference when we activate these in the morning versus in the late afternoon. But we haven't seen that effect. So
Okay.
And
that actually leads
very
into
difficult. I mean, we're working. I mean, you need to have very reproducible data to get
absolutely
statistics on that.
And then that actually leads into another question about your sleep study from one of our former undergraduates, Alan Nee, who is now at Johns Hopkins. A technical question for you, Patrik. What method of sleep deprivation was used, and was the trap paired with any noxious stimulus, or was tamoxifen given after sleep deprivation?
It was not paired.
Okay.
So it's a treadmill. So they basically walk on a treadmill during the day, and I mean the treadmill has if they try to jump off, they get a small electrical shock, so they quickly learn to do that.
To stay on the treadmill.
But I mean that I have to say, I mean the animals have been trained, and I mean we've also put them on a treadmill, nighttime as well as daytime. So it's not actually the exercise, and the treadmill moves very slowly. I think we have the right controls there.
Yeah, just enough to keep them awake.
Can I make a comment here, Harrison, on this?
Sure.
Also, the sleep deprivation studies are fabulous, Patrik. One of the things that we'd shown is that if you give a kappa opioid receptor agonist to animals, it produces profound sleep disruption. And this actually was kind of interesting because in very early old studies, that were done, unethical studies actually, that were done in I think in prisoners actually, where a kappa opioid receptor agonists were given to humans, again, they produce very profound insomnia. And so one of the things that might be kind of interesting is the kappa opioid receptor expressing cells that had been postulated to mediate descending inhibition. It sort of kind of fits a little bit with your model, not exactly, but I think it's possibly something that is relevant.
That's a great point, Frank. And then, did you want to say something, Patrik? Okay, I think this question was directed towards Kirsty. Thanks for the wonderful talks. Most of the models of QST or CPM integrate nociceptive input from the limbs or periphery. Do we expect a different mechanism of descending modulation in relation to visceral pain? How can we integrate like the NTS, the primary afferent nuclei for the vagus nerve, to brainstem circuitry involved with, like what you mentioned, the locus coeruleus, the RVM and such?
So there's definitely clear differences in the central processing of visceral versus somatic stimuli, but a common role for descending modulation by brainstem activity and mediating those evoked pain responses. I know that many years ago, Tony Dickinson and Shafak Sikandar did lots of work looking specifically at the role of modulation with visceral pain in models using colorectal distension, for example. I don't remember the ins and outs of those studies, but I would definitely recommend that someone go and check out those authors combined, because they've got lots of studies on exactly that.
Awesome. Thank you, Kirsty. That's a great point, and thanks for directing us to that relevant literature. I think that was the last. One more question. Talking about dopaminergic neurons makes me think about serotonergic neurons, which we know are involved in depression. How do low levels of serotonin affect pain modulation? I think that was not really directed at anyone in particular.
Perhaps
but
check out Tony Dickinson's back catalogue.
Right.
He's got more papers on the impact of serotonin on pain modulation. You know, if you want to get that basic fundamental physiology, I'd say that was a really good place to start.
Great suggestion, Kirsty. Thank you.
And serotonergic agonists are used for treatment of migraine, so you could think about it that way too.
Also, great point, Frank. Thank you. And so I believe that was the last question from the audience. And with that, I think we're a little bit over. So I'd like to thank the panelists. You all gave amazing talks. I really appreciate you all taking the time to be with us today and for supporting the work of the Pain Research Forum. And hope you'll all log in next time for our next webinar next month.
Thanks, Harrison.
Thanks, everyone.
Thanks, everybody.
Bye.
Bye-bye. Bye-bye.